Good morning all. I am Mrs. Kokila. Today we are going to see about deoxyribonucleic acid DNA. In this video we are going to see about nucleic acids, types of nucleic acid, structure and composition of DNA, Watson-Crick model, types of DNA and functions of DNA. What are nucleic acids? Nucleic acid are polymers that consist of nucleotide residues. Nucleic acids are located in the nuclei of cell and they are hereditary determinants of living organisms. Nucleic acids are composed of elemental composition, namely carbon, hydrogen, oxygen, nitrogen and phosphorus. Types of nucleic acids. There are two types of nucleic acid, DNA and RNA. DNA, it is deoxyribonucleic acid. RNA, it is ribonucleic acid. DNA is found in the nucleus with small amounts in mitochondria and chloroplast. It is the carrier of genetic information, whereas ribonucleic acid or RNA are found throughout the cell and it forms an intermediate in the expression of genetic information and other diverse roles. Let us see about the structure of DNA. In the year 1953, Watson and Crick proposed a structure for DNA after observing an X-ray diffraction image of DNA taken by R. Franklin in the year 1952. According to them, DNA is a polymer of deoxyribonucleic acids and it is the carrier of genetic information. DNA, it is a polymer of deoxyribonucleotides as shown here and it is the carrier of genetic information. Let us see what a DNA is composed of. Composition of DNA. There are three components of the DNA, that is a 5-carbon sugar called deoxyribose sugar, a phosphate group and a nitrogenous base. This nitrogenous base, it is a nitrogen containing ring structure which is responsible for complementary base pairing between nucleic acid strands. Now let us see the difference between nucleoside, nucleotide and nucleic acid. As seen in the previous slide, nucleoside, it is composed of sugar and base. Sugar and base together is called as nucleoside. When a nucleoside is added with a phosphate group, it is called as a nucleotide. And hence nucleotides, it includes phosphate, sugar and a base. And each monomer of nucleotide will be linked by phosphodiester bond forming a nucleic acid. Hence nucleic acid is a polymer of nucleotides. You can see a picture of the structure of a nucleotide where nucleotide is composed of a phosphate group, a sugar and a base. A phosphate group, a sugar and base linked by phosphodiester bond with a neighboring nucleotide. Coming to nitrogenous base. Nitrogenous bases are categorized into purines and pyrimidines. These are heterocyclic bases. There are 2 purines and 3 pyrimidines, out of which adenine and guanine, they are categorized under purine, whereas cytosine, thymine and uracil, they are grouped under pyrimidines. Adenine and guanine exist in both DNA and RNA, and among the pyrimidines, cytosine exists in the DNA and also RNA, whereas thymine exists only in the DNA and uracil exists only in the RNA. Now let us move to the DNA structure as proposed by Watson-Crick model. Watson and Crick proposed that DNA is made up of 2 strands that are twisted around each other to form a right-handed helix. The two DNA strands are anti-parallel such that 3' end of one strand faces the 5' end of the other strand. The 3' end of each strand has a free hydroxyl group. So here you have a 3' end, and in other strand you have here 3' end. This 3' end has a free hydroxyl group, while 5' end of each strand has a free phosphate group. The sugar and the phosphate of the polymerized nucleotide forms the backbone of the structure, whereas the nitrogenous bases are stacked inside. In the structure, you can be able to see clearly that the sugar and phosphate residue form the backbone, whereas the bases are stacked in between. These nitrogenous bases on the interior of the molecule interact with each other, base pairing with each other. So diffraction patterns of DNA infers that there are about 10 bases per turn in the DNA. The asymmetrical spacing of the sugar phosphate backbones generate major groove where the backbone is far apart and a minor groove where the backbone is close together. So these grooves, major groove and minor groove, are locations where proteins can bind to DNA. And the binding of these proteins can alter the function of the DNA, regulate the replication, or regulate transcription of DNA into RNA. Coming to base pairing. Base pairing takes place between a purine and pyrimidine. In a DNA, adenine pairs with the thymine as these two bases are complementary bases, whereas cytosine pairs with the guanine, which are complementary with each other. So in this figure which is shown here, adenine pairs with the thymine and cytosine pairs with the guanine. So these base pairs are stabilized by hydrogen bonds. There are two hydrogen bonds exist between adenine and thymine, whereas between guanine and cytosine, 3 hydrogen bonds exist which stabilize them. Now let us see about the types of DNA. There are 3 different types of DNA, namely A form, B form and Z form. We will see one by one. aDNA. aDNA, it is a double-stranded helical structure and it is much wider or flatter than bDNA. And this aDNA is one of the possible double helical structure which DNA can adopt along with other 2 biologically active helical structure, that is B form and Z form. And aDNA, it is a right-handed double helical structure and it occurs only in the dehydrated sample of DNA, such those used in the crystallographic experiments. Second type of DNA is bDNA, which is described by James Watson and Francis Crick. It is commonly formed DNA. It consists of 2 helical polynucleotides which are coiled around a common axis. These 2 helices are wound in such a way to produce 2 interchange spacing or groove, that is major and minor groove. Major groove is quite wider, whereas minor groove is narrow. The width of major groove is about 12 angstrom with a depth of 8.5 angstrom unit, whereas the width of minor groove is about 6 angstrom and depth of 7.5 angstrom unit. These grooves provide enough surface with which the proteins, chemicals, drugs can interact. Third type is zDNA. It is one of the many possible DNA double helix structure, which is a left-handed double helical structure which winds to left in zigzag manner. That's why the term is zDNA. And it was discovered by Rich, Northern and Wang in the year 1984. It has antiparallel strands as like bDNA and it is quite longer and thinner compared to bDNA. Now let's move to the functions of DNA. DNA stores the information which is needed to build and control the cell. The transmission of the information from the mother to daughter cell is called vertical gene transfer, and it occurs through the process of DNA replication. DNA is replicated when a cell makes a duplicate copy of its DNA, and then the cell divides and results in correct distribution of one DNA copy to each resulting cell. DNA can also be enzymatically degraded and can be used as a source of nucleosides and nucleotides for the cell. So in a nutshell, we have learned about nucleic acids, structure and composition of DNA, Watson-Crick model, types of DNA and functions of DNA. Hope you understood. Thank you.